Now that we've created a text file of our 130 genes with its associated fold change, again, we use the sum of all the fold changes for the 17 different comparisons. We can now import this list into Bayes-based correlation engine. Again, Bayes-based correlation engine takes into effect the direction of the change, and not only that, but the magnitude as well. So in order to import our data, we're just going to click on this button on the left, import your data. It's a pretty simple process. We can name this study anything we want. I can call it cluster 1 if I would like. I can also select the data type. These are all RNA expression studies from humans. And then I just choose my file here. Cluster 1, I've got that right here. Hit the next step. As you can see, it recognized our gene set here. But now we have to tell it what this file is. This is fold change, and it's in the plus minus mode. But again, you can choose several different ways to display the fold change data. I'm going to click that, go to next step. This is just telling me kind of a summary of my study, and it recognized all 130 identifiers that I put in. So this was actually better than String and Pathway Studio. I'm going to hit the next step. This is where you can actually put tags on it, so other people might be able to find it. We could put kidney cancer here, or any other kind of tags that we might want to put in to not only for us to find it, but other individuals. And I can just hit finish here, and it will start the import. This usually takes a while. Obviously, it has to go through a lot of data. So I have already imported this list, and we can find that here at My Data. Now that we're on this window, we can select our gene list. I put it in my folder here, but you can see there's some predetermined folders already in the My Data part. These are things that were put together by Illumina. Those are kind of interesting to browse if you want, but for this demonstration, I'm going to go right to my gene list. Here is my gene list, cluster 1. I click on that, and I'm going to click on this red window. This is my quick view window. So once I click on this, the software correlation engine will now basically direct all its bioinformatics on my list. So it's going to take my list and compare it to everything that they have. So I'm just going to click on my quick view, and now we're in another window. Given down below are the 130 genes that we imported into Correlation Engine. Again, we not only imported the gene symbols, but we also imported the sum of the fold change across all 17 different comparisons. And as you can see, Illumina has ranked these genes based on this fold change that we gave it. So our number one ranked gene is topoisomerase 2A. If you remember back to our string demo, when we were using that to connect the genes, topoisomerase 2A was the most connected gene in our network. And now it's showing up at the top of our list. So I'm not in papillary renal cell carcinoma isn't my normal field, but if I was researching aggressive forms of this disease, this might be a very good target for me to look at. And you can see Aurora kinase A is here. A lot of these are just rock stars of cancer. And if you look them up, sure enough, they are not only diagnostic, but probably indicative of the type of cancer you have in many different other forms. So right now, what we're going to do is, we're going to use these tabs up here to now investigate this list. And the first tab that everybody loves is pathways. So that's what we're going to do first is just like we did with the other software, String and Pathway Studio. Let's look at the pathways that are enriched in these 130 genes. Given down below are some of the pathways that were overrepresented in our 130 genes that were overexpressed in our more aggressive form of cancer. And again, you can, just like with the other software, String and Pathway Studio, they're all kind of mining the same gene ontology databases. So they're going to produce similar results. And again, we're seeing a lot of genes involved in cell cycle or cell division. And these p-values are incredibly small. We can look down here, cell division, chromosome segregation. I mentioned this before when we were looking at string. A lot of things associated with the kinetochore or the spindle poles. We can look down here, poli kinase signaling events. So that might be another pathway associated with these genes. Aurora B signaling. I mentioned aurora kinase A a lot. Look down here, genes involved in cell cycle. Checkpoints, that might be something that would be interesting to look at. Given here, there are, again, our genes. We have 130 genes that we found upregulated in our more aggressive cancer type. There's 117 genes associated with cell cycle checkpoints. 10 of those are in common. The odds of that happening are 1.7 times 10 to the negative 12th. And if we wanted to, we could grab all these genes down here and export them in a list and look at those as well. What I really like about, not only can we look at pathways, but we can also look at regulatory motifs. That might be something if we're looking for, say, a transcription factor that might be affecting a lot of these genes. I see E2F transcription factor in a lot of cancer data sets that I look at, and a lot of transcription factor binding sites that are unknown. So we don't know the transcription factor that's binding to, say, this region that seems to be overrepresented in these genes. So again, this is something we can use to maybe try to get to the upstream regulators in our list. Another good category here are positional gene sets, and I see that a lot in cancer. You see a lot of copy number alterations. So a lot of times when you'll see groups of genes in the same area of the chromosome, this might be something that you might be interested in. We can apply this filter. And as we look down here, the first one is chromosome 17q23. And if you remember back, our number one ranked gene was topoisomerase 2, which was at 17q21, which is very close. There's 4 genes associated here, but if you actually look down, we will see, there it is, 17q21. And here are the genes that are in that chromosomal region, and the odds of getting all those genes elevated or associated in our gene list by random chance is incredibly small. So again, maybe this is evidence that we're getting amplification in this type of tumor at around chromosome 17q21 to say 23. Again, just other things you can look at. But what I want to stress here is that basically what we are doing is mining text. And so the next tab I'm going to show you is the Curated Studies. And what we're doing with this tab is we're actually using our list to look at comparing it to other gene lists in all the different studies they have. What does our gene list most look like? And again, we have the fold change in there, so it's taking the ranking and the direction of the fold change into account. So let's look at Curated Studies.